In this lecture, we're not only going to discuss the roles of DNA and RNA, which are our building blocks of genetic material, but also how the chemical structure of genetic material impacts its function, and extend it to research implications. The history of DNA starts with the scientists Oswald Avery, Colin MacLeod, and Macklin McCarthy, pictured to the right. They worked with Streptococcus pneumonia, which, as the name suggests, is the bacteria that causes pneumonia. The scientists were carrying over work from another famous scientist, Frederick Griffith, whose experiments demonstrated that bacteria can change their form and function, a process that he dubbed as transformation. But he wasn't able to pinpoint the source of this transformation. To find the source of the transformation factor, the scientists started with a colony of S-type bacteria that Griffith had used in his experiments. This smooth bacteria was deadly compared to the rough R-type bacteria strain. In his experiments, Griffith had proven he could convert the R strain bacteria into the deadly S strain bacteria in mice. However, the source of the transformation was unknown. Therefore, the three scientists performed a controlled experiment. Using heat to kill the S strain bacteria, they divided the culture of S strain material into three new cultures. One of them contained a proteinase, which denatured proteins and ensured that no proteins were in solution. Another contained RNase, which denatured RNA molecules and ensured that no RNA molecules were in solution. And a final contained DNase, which denatures DNA and ensured that no DNA was in solution. Upon adding R strain bacteria in each solution and incubating, two of the three cultures, without proteins or RNA, demonstrated both R and S strains, and thereby the passing down of genetic information, while the other, without DNA, showed only the R strain. This proved that DNA, not the proteins or RNA, is the genetic material that passes traits to the next generation, and it was a landmark experiment in the field of genetics and biochemistry. So in diving into DNA, let's first talk about its structure. DNA consists of monomers called nucleotides, which consist of a nitrogenous base, a deoxyribose sugar group, and a phosphate group. They are held together by a phosphodiester bond at the 5 and 3 carbons of the nucleotide, as seen highlighted in yellow. There are 4 major nucleotides in DNA: guanine, cytosine, adenine, and thymine. They are categorized by the number of rings they contain. Adenine and guanine are purines with a double ring structure, and cytosine and thymine are pyridamines with a single ring. The interactions of these nucleotides becomes very important to helping us understand DNA structure and function. So now that we've introduced the nucleotides, we can discuss how they interact, and that brings us to a familiar term, hydrogen bonding. Perhaps you've seen this in other classes taken before, but guanine pairs exclusively with cytosine, and adenine pairs exclusively with thymine in DNA. To note, guanine and cytosine pairs have 3 hydrogen bonds, seen here, while adenine and thymine only have 2 hydrogen bonds, which do have consequences on stability and function of DNA molecules. These are also known as Watson-Crick pairs, named after 2 other famous scientists who won the Nobel Prize of Medicine in 1962. They used X-ray diffraction, shown to the right, to show the structure of DNA. There are 2 additional notes for the shape of DNA from the research. From a top-down view, DNA turns in a clockwise spiral, and the 2 strands of DNA actually run in opposite directions from one another. One strand runs from the 5 to 3 carbon, and the other one is anti-parallel to that, running in the 3 to 5. This directionality plays an important role in how DNA is replicated and recopied for transcription, which will be discussed later. So DNA is arranged by nucleic acids stacking on top of each other, which is known as base stacking. From the previous slide, it's clear that guanine and cytosine have stronger base stacking forces due to the greater number of hydrogen bonds between them as compared to thymine and adenine. Base stacking and the inherent hydrogen bonds play a critical role in the stability of the DNA structure. For example, areas with long runs of guanine and cytosine pairings are more resistant to denaturing. Base stacking forces that hold DNA stability are also impacted by adjacent nucleotides, not only the ones across from each other. These forces are primarily van der Waals forces and electrostatic forces, and the combination of all of these keeps DNA as stable as it is. We talked before about the double helix of DNA, which is an extremely stable and protective form for the DNA to be in. When DNA is allowed to denature, perhaps due to strong alkalis, heat, or chemicals like DMSO or formamide, DNA unfurls and exposes the double helix, allowing for nucleotide bases to absorb specific wavelengths. You can see on the plot on the right, a single strand of genetic material maintains a similar curve for absorbance, even when the temperature is increased. But the curve for a double-stranded material shifts to the right with greater absorbance at higher temperatures. When performing experiments like this, it is also important to consider the melting temperature of DNA, which is normally between 50 to 100 degrees Celsius, depending on factors such as the bonds within the DNA, the solution the DNA is contained within, and the length of the DNA. And speaking about the solutions, salts typically stabilize DNA because the cations in the salt will neutralize the negative phosphate backbone of DNA, reducing electrostatic repulsion of the double helix. There are a few other properties of DNA worth discussing that impact its function. DNA is groovy in that there are short and large undulations in the DNA structure that determine accessibility of DNA for gene transcription. The wider the groove, the greater the accessibility for transcriptional binding and gene expression. DNA is also coiled, making it easier for it to be stored and protected in the cell. During transcription, DNA is unwound and negatively coiled using enzymes called topoisomerases. Topoisomerases are proteins that regulate the coiling of DNA. Specifically, topoisomerase will break the DNA strand at specific points to reduce torsional stress and allow transcription to occur. Topoisomerases are extremely critical to opening up the DNA strand to allow enzymes like DNA or RNA polymerase to enter and to start generating respective strands. In this example, you can see that once the coil is unfurled completely, RNA polymerase can move onto the strand and then start adding corresponding nucleotides one by one. As an important note, we'll talk about this later, but RNA does not use thiamine, but uracil. You can see that using the bottom strand as a template strand, we can generate an RNA strand with RNA polymerase that is nearly identical to the coding strand, except instead of using thiamine, we have uracil. So now that we've introduced RNA in the last slide, let's dive into what RNA is by comparing and contrasting it to DNA. RNA and DNA are both crucial for life. While DNA is the primary carrier of genetic information in an organism, RNA has many forms and purposes. Structurally, RNA, as in the name, contains ribose instead of deoxyribose, meaning that the ribose has an extra oxygen atom at its 2-carbon in a hydroxyl group, rather than just a hydrogen. Within the nucleotide bases on the ribose sugar backbone, uracil will replace thymine. Probably the most important property of RNA is the fact that RNA is single-stranded, making it less stable than DNA, and referring to temperature properties about the genetic material as seen before. Since RNA is single-stranded, it also means that it does not necessarily have the equal numbers of Watson-Crick pairs as seen in DNA. In doing analysis of RNA, it is important to also know that alkaline hydrolysis of RNA can break RNA strands into smaller pieces of mononucleotides, which we'll discuss in lab techniques later in the presentation. There are many types of RNA, some which you've certainly heard before. We'll focus on 3 of the most important ones for protein synthesis. Messenger RNA, also known as mRNA, is a type of RNA that transfers genetic information transcribed from DNA to ribosomes to generate amino acid chains that eventually become proteins. mRNA is well protected as it makes its way to ribosomes with a modified guanine cap at its starting 5-carbon end and a strand of several adenines at its tail to protect it from degradation, also known as a poly-A tail. Ribosomal RNA provides scaffolding for ribosome assembly that is crucial for translation. Transfer RNA is the bridge between mRNA and amino acids. Sets of 3 nucleotides on mRNA correspond to sequences on the tRNA molecules, and the tRNA molecules are carrying corresponding amino acids that eventually will link into growing peptide chains that will become primary structures of proteins. We've talked about building proteins and genetic material, so now's a good time to talk about how to break them down. This is a full circle to the lecture, as we started with the Avery, McLeod, and McCarthy experiment, in which they used nucleases to prove that DNA was the source of genetic material that passes traits onto the next generation. Nucleases are enzymes that degrade DNA and RNA. Specifically, deoxyribonucleases degrade DNA, Ribonucleases degrade RNA, and there are even some nucleases that can hydrolyze both. There are also enzymes called endonucleases that break down the phosphodiester bonds of DNA molecules to produce new 3 and 5 carbon terminals. This then allows scientists to potentially add or remove genetic information. By using restriction endonucleases, specific sequences of DNA or RNA can be cut or modified for research purposes. Probably the most common type of endonuclease discussed is CRISPR-Cas9, a naturally occurring endonuclease that bacteria uses to take a small piece of viral genetic material and insert it into their own DNA to remember the virus, these areas being called CRISPR arrays. Upon an attack from the same virus, the bacteria, now armed with that knowledge of the virus, can produce RNA segments from CRISPR arrays to attach onto the viral DNA. This tags the viral DNA and recruits Cas9 or other proteins to destroy the viral DNA. This process has been adopted by scientists in that they create short RNA guides that attach onto a cell's DNA and allows a Cas9-like type enzyme to cut at that site. Once cut, the scientists can take advantage of a cell's own repair system to add or modify DNA to change expression. Finally, let's talk a little bit about lab techniques to understand DNA and RNA. Northern blots look to analyze RNA starting with RNA isolation, detecting certain sequences by separating them via gel electrophoresis using their relative size. After separation, the RNA fragments can be transferred out of the gel and onto a membrane with luminescent probes or tags. A similar process is done for a southern blot, except a southern blot uses DNA and requires DNA extraction and enzymatic digestion to prepare the sample prior to gel electrophoresis. An example of where this might be useful is cancer research. Overexpression of a gene compared to a normal expression can be shown through a northern blot. If the result of a test demonstrates a darker band at a certain kilobase value, kilobase being the unit of weight or size of the genetic material, as compared to normal cells, then there is likely malfunctioning of genetic expression. So with that, let's get into some questions. For question 1, which of the following sequences corresponds to the mRNA sequence of the coding sequence? Rather than take the template sequence and then working out the mRNA sequence, we can remember that the coding sequence is going to be the same as the mRNA sequence, with the exception of substituting uracil for thiamine. So the answer in this case is C. For question 2, which of the following hypothetical DNA strands would have the highest melting temperature? If we think back to the nucleotide forces from base stacking, we'll remember that the GC bonds exhibit more hydrogen bonding than AT bonds, which makes them more resilient to denaturing at higher temperatures. Therefore, it should be the strand with the most GC Watson-Crick pairs, and strand B will have a higher melting point than strand A. Which of the following is false regarding the differences between DNA and RNA? For this question, we have to think back to our discussion of how these differed. RNA and DNA certainly have different sugars in their backbones, and while RNA is single-stranded, DNA is double-stranded. Due to that latter fact, the nitrogenous base pairs are not necessarily equal for RNA. So by process of elimination, and knowing that RNA uses uracil and not thiamine, both which are pyridamines, the answer is B. Question 4. The following protects DNA against denaturing, except which of the following? So we know that base stacking and hydrogen bonding between nucleotides provide stability to DNA molecules, so we can eliminate those options. Increasing cationic environments promote stability of DNA with neutralizing interaction of positive ions and the negative phosphate backbone. However, a low pH environment can disrupt hydrogen bonds of DNA, causing breakdown. Therefore, the answer is B. Finally, let's do a lab-based question tying in some testing from the first unit of this course. A biomedical researcher is studying a rare genetic disease. The researcher's current experiment focuses on understanding the disease at a gene and protein level to identify potential targets for a therapy. She has the following methods to use: northern blot, southern blot, western blot, and mass spectrometry. If the researcher is interested in the RNA levels with gene transcription, as well as post-translational modifications to proteins, which tests should be used? So from previous discussion, we know that for RNA investigation, a northern blot should be used. And if we think back to Unit 1, the protein analysis is performed with a western blot, separating using gel electrophoresis based on several factors. Therefore, the answer in this case is A. That concludes electron nucleic acid structure and function. Thank you for your time, and thank you, Dr. Morris, for teaching this biochemistry course. These are my references.